Can we reduce methane emissions from sheep without compromising productivity? Our results suggest that we can, if we understand how these traits are connected at the genomic level. I'm Beatriz Carracelas, a PhD student from Uruguay. In Uruguay, most national greenhouse gas emissions come from agriculture, and more than half of them are enteric methane. For grazing sheep systems, methane is one of the main climate challenges. We studied merino sheep, and we measured dry matter intake, residual feed intake, methane emissions, and body weight. Each animal was genotyped, and then we performed genome-wide association analysis. When two traits share genetic control, this is called pleiotropy, but sometimes signals are simply close together, not truly shared. To distinguish these scenarios, we apply the clip test. We identify a genomic region on chromosome 10 associated with both intake and residual feed intake. This region shows strong pleiotropy, suggesting the presence of a single QTL influencing both traits. We also identify a region on chromosome 19 associated with both intake and methane emissions. The variance explained was smaller, but the signal still shows evidence of pleiotropy. Previous work from our group showed that intake and methane emissions are strongly correlated, while intake and residual feed intake show a moderate correlation. However, residual feed intake and methane emissions show no correlation. This suggests that intake is genetically linked to methane emissions, while feed efficiency is not directly associated. This creates an opportunity. We can improve feed efficiency without necessarily increasing methane emissions. Climate mitigation does not have to compromise productivity. Understanding genetic architecture makes breeding more precise and more predictable. When we understand the architecture, we can select animals that are both productive and environmentally responsible. Thank you.